Hi, Physionic. Wanted to give an introduction to what you're about to listen to. First off, the audio quality that you're hearing now is what you will get for 98% of the video. However, for the first minute and 36 seconds, something goes wrong with my audio, so it's pretty unbearable. However, after that first minute and 36 seconds, then it should be high quality audio, so you'll be able to hear everything that we have to say in crystal clear quality. The second thing I want to quick mention, this is an episode I did with Bobby Lease, who I brought on just because he introduced me to this particular paper that we're going to be discussing, looking at psychedelic drugs, LSD, DMT, DOI, a bunch of mushrooms. I mean, all kinds of stuff, and how that impacts our brain. Okay, but I get comments from individuals sometimes saying, this went way over my head. I completely get that. That is not the point of the Journal Club. The Journal Club is for those individuals that want to understand how our thought process, how somebody who's in research, how they go about breaking down a paper. So it is going to be incredibly in-depth. I do my best to every once in a while remind Bobby and of course remind myself to define particular words, but beyond that, it is going to be high-level science, which I completely get is not very conducive for actual understanding if you just want a quick answer. Obviously, by the length of this video and or podcast, however you're consuming this, you can tell that it is really long, and we take a really long time to go through all the different figures. But by the end, then we come up with some sort of conclusion, and hopefully we kind of create a snapshot. However, I also understand that you probably don't want to stick around for that entire time. I get that. This is really for those really hardcore science nerds that want to follow along with us, because I'll have the paper link that we discuss in the description box for you. However, for those individuals that just want the answer, don't worry, I will have content coming out that's specific, that is a more condensed version, a more concentrated version of what we discuss with a little bit less scientific terminology involved. And then I'll have little pieces of information as well for those individuals that just want a 2 to 3 minute snapshot of what the information is that we discuss. But this is how we got to that conclusion. Okay, so that's all I got to say. And with that said, let's jump into, and remember the first minute and 36 seconds, audio is crap. I get that, but afterwards it will rectify itself and it'll be fantastic. Okay, let's jump into it. This Journal Club. Hi, welcome to the Physionic Journal Club. If you're listening to this or watching this live, just know that we'll be taking questions if there are any questions at the end. But you're welcome to chime in, just make some sort of note of what we were talking about, so then we can refer back to that once we get to that point. So this is another special Journal Club episode, and I'm actually having a lot of fun with these Journal Clubs as I'm doing them, because they're a good way to get the real nitty-gritty science of a lot of different subjects related to in relation to different papers. So getting other individuals that are interested in particular papers or particular subjects like neurology is really exciting. It's a fun way to do things. So this Journal Club I'm doing with a good friend of mine named Bobby Lease. So do you want to introduce yourself? Do you want to talk a little bit about what your undergrad was in, and then what you're studying now, and kind of what your PhD is?
Sure.
It will be in.
So I went to undergrad college at McDaniel College over in Westminster. It's about 45-50 minutes to the west of here. I studied chemistry with a specialization in biochemistry, and my main research project when I was there was synthesizing small molecule inhibitors for PARP-14, which is, PARP is essentially just an enzyme that can run rampant in cancer. So if we can inhibit PARP, then the idea is that we can help some patients survive longer, essentially. That project went fairly well, and it's still continuing on. moving on to University of Maryland, I kind of jumped ship from chemistry more to the biology side of things and took an interest in neuroscience. I don't really know from where. But and since then, I am likely going to be joining the lab of Seth Ahmet, and he combines some cutting-edge genomic analyses like RNA-seq, ChIP-seq, some single-cell genomic stuff, with some mood disorders such as bipolar disorder, schizophrenia, and we also do some work in stem cells with some genes that are dysregulated in some of those disorders. And yeah, moving forward, we are just hoping to learn more about the brain and what happens in these disorders.
Cool. If you're not familiar with what RNA-seq is or any of the other techniques that he briefly mentioned, if you'd like to actually know about that, then let me know. Give me some sort of feedback so I can hopefully go into it. Or honestly, you can type it into Google or YouTube, and you will find many, many videos discussing it. But yeah, so today we're going to be talking about a particular paper that Bobby sent me that I think is really interesting. It changed my view on this particular topic, and I think that not very many people talk about it. Like, plenty of people talk about it in terms of, they do it, right? But they don't. Not very many people talk about the science of it, and we're talking like the nitty-gritty science of it, like what's happening on the cellular level with the addition or the use of particular drugs. And that's what we're talking about today. So this particular paper is called Psychedelics Promote Structural and Functional Neuroplasticity. And this group is going to investigate, and we're going to go through essentially a story, just like any paper is really, an extremely complex story, wherein we'll be discussing how various drugs, LSD, DMT, MDMA, shrooms, and you will definitely hear me mix some of these up because I'm not familiar with them, but just like going through the paper, I educate myself a little bit on it. So hopefully as we go through, hopefully it won't be too evident that I'm a complete noob to this stuff. But I do know some science, and I can talk some neurology. So yeah, so let's go into it. So we're gonna go through this paper. And if you're watching this or listening to this live, you're welcome to chime in on the chat. I'm not obviously going to be able to check the chat right now, but at the end I will check it. And if you're listening to this on the podcast, then I will have this paper linked, complete full access, so that you can go, and I'm going to announce each figure as we go through it. We're going to skip a few figures here and there, just kind of because it seems like I find myself in this situation all the time, these extremely just massive papers. So yeah, so you can follow along, and let's get into it. So what did they want to do? So I've got written down that they wanted to know how various drugs would impact the brain, and they made essentially a relationship to ketamine. So ketamine being, do you know anything about that? Do you want to talk about that a little bit?
Yeah. There's a couple groups at University of Maryland, actually, like the group of Scott Thompson and Todd Gold that are part of the ketamine project. So ketamine was recently, within the past couple of years, approved by the FDA as a fast-acting antidepressant. Ketamine is obviously illegal, so you do have to go to the clinic to get it, and I believe it is a nose spray. And so essentially the way that it works, without going too far into detail, is you get the spray up your nose, and within an hour, your depressive anxiety symptoms are alleviated. And this lasts for about 2 weeks, sometimes longer. Then you go back and get it again.
2 weeks, wow.
Yeah.
That's I mean, think about that. Like, you literally take a spray, and then for 2 weeks you're good. Like, that's profound.
It's crazy. I mean, the current antibiotics that we have now, like Zoloft, they take 3-4 weeks to start to work, and they only work in 2/3 of the population alone. Ketamine works in under an hour, and the part that really has neuroscience interested is that the half-life of ketamine, it's out of your system in a couple hours.
Right. Yeah.
So the fact that it's out of your system, way out of your system by a day's time, and you still get the effects for up to 2 weeks, that's what's really interesting and intriguing about it.
Mhm. And they talk about that. So they talk about, like, with the use of these drugs, essentially they want to create a parallel between these drugs and the profound impact that ketamine has. And I know in science we use ketamine for like animal anesthesia and things of that nature, but I'd never thought of it as, I never knew that it was an antidepressant of any sort. So it was really a foreign concept as they were talking about it. So so yeah, we're going to see how various drugs, again, the LSD, DMT, DOI, which we'll go into because that's one that I'm completely unfamiliar with, and see how those relate to ketamine in terms of their impact, and if maybe there is a possibility to switch ketamine out or some other antidepressant. You actually mentioned it already, that in the body of the paper they talk about how 2/3 of individuals get benefit from antidepressants, but there's a massive chunk of individuals, like 1/3 of the population, that just no antidepressants work. They just don't work whatsoever. So, and ketamine seems to have an impact for those individuals, a positive impact for those individuals. So, yeah, so I talked about ketamine. One more brief thing that I want to mention is that they talked about neuronal atrophy or depression with general depression, so, and anxiety as well. So that the prefrontal cortex, which you can literally just like point to it, right? It's like where your forehead is. Yeah. That is an area of the brain that's highly developed, and it's kind of a later stage of development for our brain in general. And it's responsible for all kinds of different things, especially thinking and planning and things of that nature, which I'm sure you know a whole lot about. But they essentially said that with depression, those individuals will start seeing atrophy in the neurons that make up that particular part of the brain. So then the study is done on those kinds of neurons. So let's talk a little bit about the method. So talk to me a little bit about these different drugs. Like, I mean, again, I know LSD, like literally the 3 letters, that's it. Like, and I know maybe it makes you see things. Like that's about it. So like, what can you tell me about like LSD? Like which drugs did they primarily focus on?
Okay, so let's go one by one. So LSD, I probably wouldn't go as far as like your typical cartoon hallucinogenics, right? When most people take LSD, they're going to see patterns among things. So like they're not going to see a visualization of a person standing there unless they take an extreme dose of it or something like that.
Yeah.
So most of the time you'll see patterns, you'll see things be described as breathing, essentially. And on top of that, you can sometimes get inside this loop in your mind, and you'll just kind of keep running around this loop, and you won't realize it until you actually realize it. Then you're like, wow, I've been in this loop for like 10 minutes or an hour sometimes.
So you lose track of time.
Essentially, yeah. So LSD is probably one of the most common psychedelics out there. Mm-hmm. Is illegal. I believe it's Schedule 2. And that's pretty much it for LSD. Shrooms, mushrooms, psilocybin, they refer to it in this paper as psilocin. So psilocybin is the active ingredient in mushrooms, in psychedelic mushrooms. It gets converted to psilocin in the digestive system. It's just a dephosphorylation. And psilocin is actually the active compound that allows you to hallucinate or trip in the brain. So psilocin is similar to LSD, some of the effects that it will produce. It's been said to be more of an emotional, more type of a spiritual experience. A lot of people who do take shrooms do describe it that way, whereas LSD can be more of a uplifting, kind of energetic experience. MDMA is commonly known as a party drug. The pure form of MDMA is typically called Molly on the street.
Okay.
When it's kind of laced with caffeine and some other things, it's typically referred to as ecstasy. Okay. So ecstasy and Molly are generally the same thing.
Yeah.
It is considered to be a bit more toxic. It can cause what's known as serotonin syndrome, where it essentially just depletes all your serotonin, and then you go into a major depressive episode following the use of it. So that's something that you have to be very careful with, which is why moving forward, MDMA has its benefits. such as PTSD, for example.
Okay.
One good example, and the FDA actually recently approved this as well, is MDMA in the clinic for psychotherapy. It lowers the fear response. So it allows you to explore memories where trauma may have been,
Right.
without feeling fear of that trauma. So it allows you to really face your fear, so to speak, and alleviate it. And like I said, PTSD is probably the biggest thing that we can use MDMA for currently.
Okay. And then the other one was, I think, DMT, which
DMT.
is something Joe Rogan talks about a lot. I can say that for sure. People make fun of him for it a lot. Anyways, go ahead.
DMT is more of a spiritual drug. I don't know too much. Like, I've researched LSD, I've looked into shrooms as well, just because they are more common and we do have a lot more information on what they actually do to your brain. DMT is one of the lesser known ones, and it's supposed to be very powerful. A lot of people can describe meeting God, meeting aliens, something of that nature. So where, like LSD, shrooms, you kind of report patterns, some colors, breathing type stuff. DMT is kind of that hard psychedelic that you might think of when you hear like a trip.
Okay.
Then the other one that they talked about in this paper is one that I'm even less familiar with, and that's DOI.
Yeah,
like you mentioned.
I had no idea.
When I looked in that just briefly before this journal club, DOI is something that they call super LSD in,
Okay.
I think it was down in Australia. I really
Okay.
haven't heard of it much over here in the United States.
Uh-huh.
It's supposed to be, the effects should be similar to LSD and Molly. So MDMA and LSD, except both dampened a bit, so it's not like a full explosion, basically.
Right.
But once again, I'm not as familiar with that one.
Okay. But and I don't even know how to characterize this. Are they all kind of in the same family? Because they seem to have slightly different effects.
Right.
So, and they do mention that in the paper. I didn't fully understand that aspect though. Do you know if they're part of like different families or subgroups of particular families of drugs?
Right. So yeah, they're all considered, like psychedelic drugs, and I do think they mention it somewhere that there are a couple of different ways to classify them. You could look at the structures, which I think is in the first figure, and
Yeah.
we'll get to that, I'm sure.
Yeah, we'll get to it.
And we can talk about the pharmacophore when we do get there, but they are, a lot of them mimic serotonin, so very similar. I mean, a couple of atoms separate them from serotonin.
Yeah.
And
Right.
so that's a,
So they're very similar to one another in terms of their chemical structure, at least.
most of them, yeah. Like,
Most of them.
LSD and psilocybin, even though they produce very similar effects, are actually pretty different. And I wish I could find it right now, actually. It goes into amphetamine, tryptamine, and ergoline are the three chemical classes
Okay.
that they are divided up into. And amphetamines are amphetamines, same category kind of as methamphetamine.
Right.
Then tryptamine and ergoline, I believe LSD is considered ergoline, and tryptamine, I want to say MDMA falls in the tryptamine family.
Okay.
But either way, they all cause effects similar to one another, but different. They each have their own personality, so to speak.
Okay. Good. Which adds even more interest to the actual paper, because then they're not just testing one particular drug or one class of drug. They're trying a few different ones, and we're going to see what impact that has, unless you've already read the paper, for whoever's actually listening to this. So let's talk a little bit about the methods. We'll probably go, as we go through figures, we'll kind of explain the methods a little bit more, but they used in vivo systems and in vitro systems. So what does that mean? An in vitro system is essentially kind of outside of the body, but it's more of a cellular system. So looking at particular cells, like if you were to do microscopy in terms of using a microscope to fluoresce particular proteins, that would be considered an in vitro system. An in vivo system is within the body, so within a particular, like let's say if you've got a mouse and you do something to that mouse, that would be an in vivo system because that is a living creature that you're acting on. So they injected the drugs in vivo. They used DMT and ketamine in rats. So they used rats, and then they also used what's called primary cultured cells, where they took the cells out of, I believe it was in the rats in this case as well, and then they were able to plate them, so put them on a dish, and then see what's going to happen to those particular cells and do various drugs. They used other drugs like Ana-12, rapamycin, and ketanserin, which I've never heard of before. I've heard of the other two. I'm not going to go over those right now because I think it's probably better to go over them as we run into them. And I've got notes on that as well. And they literally only gave it to them for 10 minutes. Like, I mean, some measures they did 24 hours before or did the administration of these drugs 24 hours, and then they measured 24 hours later, and then they had a 10-minute system as well. So, and then they did microscopy, which I already mentioned, which is essentially microscopes being able to look within the cell and being able to track different proteins and things of that nature. Anything you want to add kind of on the surface?
I just want to go back on something that I said because I did find it in the paper.
Sure, go for it.
So DMT and shrooms are considered tryptamines.
Okay.
DOI and MDMA are in the amphetamine family, and LSD is the ergoline that they used.
Okay.
That's just all I want to say.
No, that's good. That's good. It's good to get things right. All right, so let's get into the results. Hopefully, if you're live, hopefully you guys can hear us because the audio is straight through the computer and not through my fancy microphones that I've got set up, which are for the actual podcast. So if you're listening to this on the podcast with crystal clear audio, just know that we took a lot of effort to make sure that was the case. Yeah, but if you are live and you can hear us, or even if you can't hear us and you have a question that you want answered that I can post later on, then you can post that in the chat, and I will hopefully get to it once we're done going through the actual paper. Okay, so let's go through the results. So I've got everything broken down. I've got a bunch of different notes on all kinds of different results. I will say I wasn't all that impressed by how they went about explaining some of these things. Like they didn't even some of the explanations they had for these results, like sometimes they just skipped over certain figures that they had and they went straight to like supplementary figures, which supplementary figures, if you're not familiar with what that is, that's essentially figures or data that they weren't allowed to put into the main body of the paper because it would have made the paper far too long, so they had to kind of truncate it down to a distilled version, kind of tell the story without going way out and just start going into all these different avenues. So that's why they've got supplemental figures. Okay, so let's go into figure 1A and B. So I'm going to lump those two together. A, do you want to talk about that a little bit? I mean, it's pretty straightforward, but...
Yeah, sure. So A is just going over the chemical structures like we were talking about a little bit. DMT is similar to... it's in the supplementary figure, of course.
Yeah.
But DMT is very similar to psilocin. So shrooms, essentially.
Okay.
And they both are very similar. Once again, just you take off those methyl groups on that nitrogen, and you just have an amine there, and you basically have serotonin.
Yeah.
So some mimic serotonin. Others, like LSD, you can just see a similar pharmacophore where you do have, the phenyl group there. You do have the NH in the second ring. Something similar to that. And then in terms of B, it's just looking at... This is actually where it starts, with the dendrites. So dendrites are essentially just projections that come off of the body of the neuron, the soma, the cell body. And dendrites are crucial to making synaptic connections. So you're not going to get any synapses, and no information is going to be passed from neuron to neuron if you don't have these dendrites. Mm-hmm. As we talked about a little bit in the introduction,
in depressed patients, they see atrophy of these neurons. So they're
Right.
not forming new connections the way that they should. And it can be described getting stuck in the same thing, like the same loop. I don't want to use loop again because LSD, but you can get in this depressive loop and you can't really break out and you can't find happiness. And some of that can be contributed to your neurons aren't making the connections that they should be making.
Okay. So yeah, so here the illustration is, and to be completely honest, it's a little silly to me. It's just like, hey, we've got a bunch of data, but we're going to just draw what we think happens, which I find kind of comical, honestly, because they've got some really beautiful microscopy, some microscope images later on, which is far more telling than any illustrations that they're going to have. But I digress. It's still, I mean, I guess it's kind of cool. So let's, you mentioned this, so dendrite. So to characterize this, a neuron, so if you think of your brain, it's filled with different cells, not just neurons, but we're going to be focusing on neurons. So neurons allow, as you mentioned, this communication between one neuron to another neuron, and then of course it allows us to have thoughts and all kinds of different things. I mean, just it's a mind-blowing area of the body, which really fascinates me, but everything fascinates me about the body. So, but you mentioned dendrites. So if we really classically think about a neuron, we can think of it as having three main sections. So a dendrite, which receives signals, a soma, or a cell body, which essentially integrates those signals. So essentially decides, okay, are we going to continue the signal or are we going to stop the signal? And then an axon, which is essentially the messenger. It sends the signal out to the next neuron, which then that neuron's dendrites would then accept the signal. And it just goes on and on and on like that. Now, in C, what they're looking at is, they mentioned this in the paper called, and I had to look some of this stuff up, some of the like terminology, dendrite arborization, which is literally just dendrite branching, which is just and you mentioned this already a little bit, but like the dendrites kind of spreading it, like there's more dendrites being created, and they go into more detail with that as well. So C, what did you get out of that? Like, did you get anything? Like, what did they do roughly, and what did you get out of it?
So C is just a quantification. It's shoal analysis, which they use a lot in
They
this
do,
paper.
yeah.
And it's still something that I'm not too familiar with.
Yeah, so I had to look that up, and a shoal analysis is a combination, and if a neuroscientist is out there, I always say this, please, if you're an expert in this particular area, feel free to jump in. I will add notes to correct things and whatnot. But a shoal analysis is looking at the length and the thickness of a dendrite or a neuron, and that's supposed to be associated with growth. I mean, if you have greater length, if you have greater thickness in combination, then you're going to have greater growth. So anyways, go ahead.
So these three graphs, essentially what they're showing is LSD, DMT, and DOI. And once again, in supplementary figures, they go on to show that Shrooms, MDMA, and the other psychedelics also do the same thing.
Uh-huh.
What they're showing is a vehicle control. So it's just a negative control, or not a negative control, but just a control.
What is a control?
A control for an experiment. So
yeah, just any experiment.
Okay. So in general, a control is something that tells you your experiment is working. So you can't, I guess that's a step ahead, but you can't say that your experiment worked if you don't have a control, because you don't know if it's because your experiment that it worked or if it's just something that happens.
Right.
So a control really allows you to differentiate between the normal and your experimental condition.
Right. Yeah, it's really simply put, that's exactly what I needed. Yeah, good.
Okay, sorry. So they do have this control here, and they just showed the typical number of dendritic crossings. Then with the psychedelics added, like LSD, DMT, they show a kind of a dramatic increase, almost a twofold increase in some cases of more dendritic crossings. And that's just going on to say, hey, these dendrites are branching out more, they're crossing more.
Right. So you're just getting, essentially think of it as just greater complexity. They're adding these drugs, and suddenly you have this just greater complexity going on with how the neurons or the dendrites are crossing with one another. And then they did, for D, they did and we can lump some of these together, like D, E, F, G, H, and I. I mean, a lot of them are just quantifications of each other, and they're just kind of separations of one another. But as a general trend here, we're seeing that the length and the width of these dendrites is growing with the administration of these drugs. I think the only exception was in primary dendrites. They showed that I believe DOI was not as effective, but the other two drugs were. So again, the three drugs that they're primarily investigating right now is the DOI, the DMT, and the LSD, which Bobby explained earlier. And also another thing is that the longest dendrite was no longer. So the maximal length of these dendrites was not changed, but more of the dendrites reached a longer stage. So the average increased, but the absolute maximum did not increase, which I thought was kind of interesting. I wouldn't have thought of that, So do you have anything to add for anything from D all the way to I?
No, that's pretty much it. Like you touched on, the length of the longest dendrite didn't change, and is surprising. So what we're seeing here is an increase in complexity, but not necessarily a change in the neuron itself, I guess you can say. Like, you're not seeing a change in the longest length, how far the dendrite can actually go. You're just seeing more get there, like you mentioned.
Yeah, exactly. Did you look up what an instar was? Because I had to.
Please enlighten me.
For figures, okay. So for figures 1, J, K, M, and N, and honestly L and O as well. But let's just look at J and M. So the first instar and the third instar, I was like, what in the world is an instar? And it turns out that those are essentially like larvae of flies. So the first instar is the first stage of a fly. And if you're an entomologist, please correct me. Like, I'm just going to keep saying that. And then the third is a more mature fly. And that's incredibly basic terms. So here they wanted to show with a newborn fly, which I know most people would probably think, well, why in the world are they testing this out on flies? Like, I know that's what people ask. Like, I've had that people ask me on content before, like, why are they looking at flies? And do you have any insight on that? Because I can certainly speak on that a little bit. But
model organisms? It all depends on what you want to accomplish. I'll just say briefly that flies have a very fast turnover rate, so they're just a fast experiment to do, as opposed to mice, which take several weeks to months to accomplish
years.
anything. To years, yeah. But if you want to go into farther detail, please.
Well, I think those are two excellent points. I think the third one that I would add is that their neurology is so simple. Like, a lot of the conserved pathways, you wouldn't think about it, but we have similarities to flies just like we have similarities to mice. And of course, the similarities increase as you go further up in the mammalian system. So obviously a fly isn't going to have as much of similarities to humans as, let's say, a mouse, and then, a primate, et cetera, et cetera. But a fly does have a really simple neurological system, and that makes it really easy. Actually, going back to your controls, it eliminates a lot of confounding variables, because you can just investigate, okay, I just have to worry about this one neuron. I don't have to worry about 30 different other neurons acting on that, or that neuron acting on those 30 other neurons, which you would with human. I mean, we are complex. Like, our brain is literally the defining feature of what makes us so incredible, to toot the humanity's horn. I mean, in all reality. So they wanted to check the first instar, the beginning of the fly's growth, and kind of a middle point, because apparently there's, I think, 5 instars. So kind of a middle point and then beginning. And what do they find? Do you want to jump into that?
Sure. So with LSD and DOI, what they're finding is that the branches, so the complexity of the dendritic branches are increasing, but the total length is not. So you're not forming new dendrites by doing this. You're essentially just changing their shape from like a long, thick dendrite. You're allowing it to branch out more, which is, again, going to get back to,
you're allowing it to form more connections. There's just more area there for it to form connections.
Yeah, exactly. And then they have representative images. If you're listening to this, check out the paper, literally figure 1, L, and O. You can see a comparison. So the vehicle is essentially a control. It's a untouched, unmanipulated neuron. And then the difference between that and the DOI and the LSD, which you mentioned, is that you start seeing this greater arborization. You start seeing these dendrites branching. So suddenly you see the complexity starts to increase already in this fly model. You're starting to see this effect. And it does not take long whatsoever.
No.
Okay, so let's jump into figure number 2. And here is where things start getting into a little bit of molecular biology, definitely some microscopy involved, and some electrical signaling, which not a lot of people are fans of, which I completely understand. Okay, so figure 2. I had to switch over to look at my other notes. Okay, so figure 2A. Here, do you want to explain this one?
Sure. So I think it is crucial that we understand really how synapses are formed.
Okay.
So you have these dendritic spines, right? So on the dendrite, you can think of them as little bubbles.
Yeah,
almost.
that's a good way to put it, for sure.
And there's tons of these little bubbles, and these little bubbles are actually where the synapse is going to be formed. So it's not going to be directly on the dendrite.
So what you are looking at in figure A is actually this the spine. So you're looking at
These little bubbles is what they're interested in this figure.
Yeah. So, and we're not talking about the actual spine, like the spine of a like your spine. You know, like people think of like your actual spine. This is a particular term for a dendrite, like you explained, where you start seeing this kind of budding that's starting to occur. So it lends further evidence that these drugs and the drugs that they looked at, DOI, DMT, and LSD in this situation, increase these dendritic spines to start creating more synapses and whatnot. So then for B, and this is where I have a lot to say, let me say, B is interesting because here they're again characterizing the morphology or the way things look. So you're starting to see this budding of these synapses, like you explained, and it can go through 4 different stages, essentially. And I think I've got that written down. Yeah, so the Philopodia, they don't get too creative with some of these. So Philopodia stage 1, stubby is stage 2, then thin is stage 3, and then mushroom, not the drug, is stage 4, and that's a mature synapse that that can actually work, that really functions fully. And there, with B, you're seeing this quantification, and you're seeing that again. I mean, it's really consistent, which is great about this paper. That LSD increases it, DMT increases it, DOI increases it. You're getting this complexity in the brain that's starting to occur from the addition of these drugs. Now, what's really interesting about B is that they're showing the distribution within X amount that they're measuring, the distribution of how much is immature, like highly immature budding, to completely mature budding. And this is where I can make an argument, if I wanted to, that these drugs could be, I wouldn't say harmful. I wouldn't say harmful because I think basically a lot of the evidence that we're going to go over and that we have is, well, I'm not going to go into it yet, but let me just say that if you look at the vehicle, the control, you have a higher percentage of mushroom or mature dendritic synapses that are formed. So the morphology is arguably more favorable for the actual communication between different neurons. However, with essentially all of these other drugs, well, with the actual drugs added, you see that there's very little of this actual mushroom. So you see far more of the philopodia, which is the first or the immature form. So is it possible that the cells are getting more complex, but it's not, it's like a useless growth or a useless sudden complexity that's occurring in the brain? I think that's a possibility. That's not necessarily what I believe, but I believe that's a possibility. Now, the argument there is that the vehicle or the control neurons, they are the baseline neurons. So they could have developed this maturity way before the treatment was even added. And then once the treatment was added, you're seeing this change in morphology, not because the neurons are any worse off, but because the drugs haven't had enough time to remodel these neurons. And in that case, then you're seeing this dramatic change again in the quality of the neuron, which could be certainly extremely beneficial, but it could also be extremely detrimental. And that's a question that comes down to kinetics then. How much time do you give these drugs? If they had measured a week later, would they have seen similar to the control levels, but much more of it? And I think that, to me, when I was thinking about that, I was like, that could be a really interesting follow-up study. Did you have anything you wanted to add to that?
No, I'm just on the same page with you. I did find it interesting that it's a higher percentage of them are in a more immature stage.
Mm-hmm.
But like you were kind of touching on, we don't know if that's because it's actually regressing anything, or if all these new spines just haven't had the chance to mature yet.
Yeah.
So
Yeah.
obviously, if you do have 10 new spines, if you had 10 and now you have 10 more, and the 10 were mature and now you have 10 immature, the proportion of immature to mature is going to drastically change. So it would be interesting to do a follow-up study. I don't know how long it takes for these to mature. I really don't.
I couldn't tell you either. I couldn't tell you either. Yeah.
It would be interesting to see down the line what actually happens. And I think that's a really important issue. And not only on psychedelics, but on like marijuana, for example, that's becoming a lot more mainstream, a lot more popular and legal. We don't know the long-term effects. We don't know the long-term effects of LSD, of shrooms. We don't know the long-term effects. So studies like this are going to be crucial to any legalization and how
Absolutely.
the government's going to continue to view these.
Yeah. Yeah, exactly. For D, well, I guess I covered B and C, but for D, they're essentially looking at two different proteins, technically three different proteins, so V-GLUT1, PSD95, and here they're looking at representative images, microscopy images. So they just want to know where is it located and how much is located based off of the intensity of the fluorescence that they're getting off that protein. And they're quantifying it then in E, F, G, and H. Do you have anything that kind of came out of that? Well, actually, before you go, let me quick jump in. V-GLUT is a vesicular glutamate transporter, so that is a part of a functioning neuron. You would need that there. Glutamate is one of the, I think the most, I want to say it's, I looked this up, I think it's the most common neurotransmitter of all, like across the entire body. So it's extremely prevalent. PSD95 is a post-synaptic density protein. And then they also looked at what's called a microtubule-associated protein, which is something that's found in basically every single cell. And microtubules allow for cells to have structure and for proteins to move from one side of the cell to the other side of the cell. So anyways, so on EFGH, did you see anything that kind of jumped out at you or anything that you found interesting?
I mean, the interesting thing is going to be the density versus the size. So in this, I guess it relates back to what we were seeing in C and B, the synaptic sizes aren't changing. If anything, you can see a small decrease in like DMT from the vehicle.
Right.
Probably not statistically significant, but it's hard to tell just eyeballing it.
Yeah.
But so you're not seeing a change in the synapse size, or really. Yeah, but you're seeing a change in the density. So you're seeing a change in the amount of molecules, the amount of proteins that are pushed right at the synapse, ready to go.
Yeah.
So a lot more of them are just ready to fire, they're ready to take off, and that just leads to more communication in your brain.
Yeah. And I think in general it kind of falls in line with a lot of other things that they've shown so far. Density increases, and for G they show that density only increases in the LSD. Honestly, if you look at those bars, I don't know, I didn't even bother to look at the statistics on this particular aspect, but like the bars just to me there's a definite distinct difference between DMT, DOI, and the control. So at least there's a trend there, I would think. But at least with LSD you see this increase in density. I'm not going to touch on I because it's more microscopy. They're showing the exact same thing, increases in complexity, things of that nature. Did you want to discuss a little bit on the frequency and the amplitude? Do you want to talk? Can you talk a little bit about what EPSCs are?
Right. So, if I can I skip to M? Because I think M's interesting.
Yeah,
I really want to
absolutely.
touch on
Which actually ties in with K and L. They mentioned that in the paper. So that's a representation of K and L. Yeah, sure,
Right.
Go for it.
Okay. So M I found very interesting. And M is just, in my opinion, it's easier to see, easier to read to the layman.
Yeah, absolutely.
Once again, not a big EPHIS guy here.
Me neither.
In terms of EPSCs, they are excitatory postsynaptic currents. So
Okay.
you're essentially just looking at. So, without going too far into EPHIS here, because I really don't want to.
Yeah.
Staying away from that for a reason. Whenever an event happens, it creates a charge, so to speak.
Right.
And that charge can be quantified. And that's what an EPSC is. It's just a quantification of a charge. So you can just use it to measure events that happen, spontaneous events, as they're
Right.
called. So looking at M, and looking at this whole thing, what I found interesting is that they see an increase in these spontaneous events. So neurons are just firing more. They're talking more at 10 milligrams per kilogram. And they're also seeing the same thing at 1 milligram per kilogram.
Yes.
And they discuss in the paper itself that it is predicted that and once again, these are in just plated cells.
Yeah.
So it's hard to. But they say that 1 milligram per kilogram doses are predicted to be sub-hallucinogenic. So there's this thing called microdosing. And it's where a person the most common method would be to take a microdose or a very small dose of LSD or shrooms, your choice. Day 1, then you have 2 days off, then you have another day on. And people express, especially with LSD, increased focus. Some people have said that it's gotten them off of their ADHD medications even. People are generally happier. They have more energy, and they're just they claim that some of their depressive symptoms are relieved because they're taking this microdose. They go to work, they go to school, whatever they want to do on this drug. It is sub-perceptual, so you don't get hallucinations.
Right.
You wouldn't be able to tell you're on a drug. You would just go home at the end of the day and be like, that was a good day.
Yeah.
And then the effects, part of, as they're discussing in this paper, the effects don't last just the length of the drug. You'll see some of the effects into the next day. And then the third day is typically considered more of an off day
then you're back on it again. So people, I think this figure alone makes a strong case that microdosing should be looked into more. Like I said, it is a sub-perceptual level, so you're not getting hallucinations. So you're not going to be able to tell that a person took a little bit of LSD or a little bit of shrooms that day if you were just communicating with them.
Yeah, right.
And I think that's, in terms of future therapies, I think that's promising, that we do see so many, more spontaneous events even at that sub-perceptual level. I think that's exciting.
Oh, it definitely is. I think that's something we can talk about near the conclusions again, just to reiterate it, because it's absolutely. I mean, it's almost like you could take them like vitamins.
Yeah, honestly.
Like I need to take my multivitamin, my fish oil. Let me take my DMT. Yeah, absolutely. And I think you jumping on M specifically is important, because you can actually really see the differences with K and L. It's hard to tell the differences. Especially L, like the differences in amplitude. I was like, okay. I mean, I see it, but it's you have to use a microscope just to be able to see the differences. J, I just want to quick mention that before we move on. J, they looked at ketamine specifically, which is the thing that they had mentioned at the beginning, which is used, and you mentioned this in detail about how that's a really powerful antidepressant and it helps a lot of people. And they wanted to stack that up against DMT, and I guess presumably did maybe slightly better, but DMT was right in that realm. So it also increased this complexity in the dendrites, just like ketamine. So you're seeing that crossover between the two drugs.
I really wish that they would have this figure with LSD, with shrooms, because I don't typically hear of people using DMT to microdose or something along those lines.
Talk to Joe Rogan.
Maybe I should.
But yeah, I mean, I would have definitely been interested to see these EPSC events in LSD or shrooms. And I'd like to know if they are along the same boats as DMT and how they stack up against ketamine. I think that would be interesting, but they don't.
Yeah, they failed in that regard. They didn't have a whole lot of data on ketamine, which I was a little disappointed on, and I'll touch on that as we get to the conclusion. Figure 3, here they're looking at BDNF, which is brain-derived neurotrophic factor. Now, this is something that I've looked into a little bit before, because it's actually something that exercise physiologists push, because exercise dramatically increases BDNF levels, and that has been especially correlated and really even causative of increase in neuronal growth. So here they're looking at BDNF, which is a really powerful brain stimulating. It's essentially seen as this like overarching great thing to have in high quantities in your brain and in your neurology in general. So here again they're using a shoal analysis, which we talked about earlier, looking at the max thickness as well, the thickness and the length of the dendrites. And they're adding different quantities of BDNF. So they just want to know what happens when you add BDNF, completely independent of the drugs, because they're going to bring in the drugs later. So with an increase in BDNF, so they added 0.5, and I don't see the actual values. I guess it's nanograms. So they added 0.5, then they went up to 5, then they went up to 50. So kind of a tenfold increase from one to the other. And with 5 already, they saw increases in the thickness and the length of these dendrites. So, and this literally just plays straight into all the other literature that already says BDNF, good stuff to have, essentially. And for the max thickness and max length, with the drugs earlier, we didn't see any changes, but with BDNF at really high doses, 50 nanograms, that you do see this continued increase in the thickness and length of the neurons. And C is just a plot of that, so we can just go over that as well. Do you want to touch on D then? Because that's where they introduce the drugs?
For sure. And once again, they're using only DOI here, which is
problematic because they're not showing all the other psychedelic drugs, but that's
right.
Okay. So what they show is that when they do add BDNF, you do see a dramatic increase in the number of dendritic crossings,
Yes.
just like we were talking about back in
Right.
figure 1.
Right.
When they add DOI, they see the same thing.
Yes.
Like that dramatic increase. When they add them both, they don't see a farther increase. They see the same increase as they see when they only add BDNF and when they only add DOI.
Yeah.
So that's interesting. That essentially says, okay, BDNF and DOI, they're likely using the same mechanism, because we're
Yes.
not seeing any increase when you put them both together.
Right.
So the thought is that the psychedelic drugs use BDNF and some farther pathways to help promote plasticity. Because BDNF, like you were talking about, that's why they say good exercise and wanting to exercise can really help you with depression, because it, like I said, increases the levels of BDNF. BDNF increases plasticity, and plasticity is just referring to, a neuron's ability to change over time, essentially.
Right.
So it's interesting.
It absolutely is, and you hit the nail on the head. That's exactly what they're trying to show here, that with BDNF and DOI, you're not seeing this additive effect. So you can choose one or the other, and they're gonna go through what presumably is the same pathway. I've got a few things to say on that, but that's going to be figure 4. They're going to actually start looking at, like, okay, we have this evidence here that there's no additive effect. So, DOI could be binding to the same spot that BDNF binds on the cell, and then it's going through the same signaling cascade within the cell, meaning that you have a molecule that binds to the outside of the cell, then the receptor will activate other molecules inside the cell to start moving to other areas, and then start promoting whatever it needs to promote, or inhibit whatever it needs to inhibit. In this case, BDNF would then promote the growth of those neurons. So DOI and BDNF, this lends, and like you said, that DOI most likely works through a similar mechanism, that's why you don't see this increase, because the mechanism that BDNF works through is completely saturated, essentially. Like you're not going to get more. It's not moving through a separate way. It's like using one highway to get there, and you're filling it with cars, and you literally can't get to your destination by any other means. It's just this one highway. So once it's packed, it's packed. Like you're not going to be moving any quicker. You're not going to be getting to your destination with any more precision. Then for E, honestly, it's just a quantification of what you already talked about. G, here they're showing gene expression of BDNF. So because our cells are able to create this BDNF, you have this expression, you need this expression of BDNF. So what do they show here? That there's no added expression. You don't see a change in expression in BDNF, which is fascinating to me. And I mean, immediately my brain started thinking of all these different scenarios of what could be happening, where you're seeing this, where you're seeing these drugs potentially having an impact, but the gene expression to then create the protein BDNF is not upregulated. But then they look at H, they actually show the protein levels, and they show that it's not significant. What did you think about that? When you look at that, when you look at H, and you compare that to the control.
Yeah, I don't know how they're saying it's not significant. DOI alone is literally an entire magnitude difference.
Is. Yeah.
So
well, all of them are. I mean,
Yeah.
look at the standard errors. I mean, I think the standard errors.
Yeah, so I don't know where they're coming up with not statistically significant, because at least on the graph that they present, it's very significant.
Yeah. It absolutely looks like it.
That
so
is interesting.
did you have any thoughts on G and H kind of in combination?
I'm curious to know why. You
Yeah.
know, what's happening? Is it stabilizing the mRNA? Is it allowing it to... what are these drugs doing to the BDNF transcript that allows it to produce more protein?
Right, exactly. And for figure 4, they do exactly that. They start investigating those kinds of questions. So we mentioned that BDNF is really important. It's really helpful, all these different things. But it binds to the outside of the cell somewhere. And that outside of the cell is through a receptor. And that receptor is called TRKB. And BDNF binds to that, and then it leads to this, like, telling the cell, okay, go, grow, do this thing. And so what they wanted to know was if they add one of the drugs, so not one of the psychedelic drugs, but one of the clinical drugs to stop that from occurring. So when they wanted to study BDNF and they wanted to study these drugs and how they potentially work through a similar mechanism. So how do you do that? Well, you knock out that mechanism. You try and destroy it so that, and then see if you still see that effect. And so they add this drug, Anna 12, and Anna's not a very nice person. And it's, I guess it's probably too much information, but like it antagonizes. It's a non-competitive antagonizer, but it antagonizes TRKB. And so do you want to go into like what did they show?
Yeah, so when they added this Anna 12, this TRKB antagonist, this inhibitor, I guess you could put it as,
right,
they showed that even with the drug,
like LSD or TOI or DOI rather, you don't see the same number of crossings, dendritic crossings.
Yeah.
So you're essentially everything that they've shown up to this point, they show an increase in dendritic complexity. When they inhibit this one receptor, it loses all of that complexity.
Yep.
Well, it's not that it loses it, but it never gains the complexity,
right,
should I say.
Right.
So it's not direct evidence because other things could be going on, but it's strong evidence to say that TRKB is and that signaling pathway is very important for the downstream effects that we see with these types of drugs.
Yeah, absolutely. And you said it perfectly. Now, did you think anything special with DMT? So you mentioned DOI and you mentioned LSD. Both of those are dampened to control levels, like as if they don't exist. When you add them and you add this inhibitor, that inhibits the BDNF pathway. So what did you think of the DMT figure?
I don't know.
So for me, when I looked at that DMT figure, you're seeing this reduction, this inhibition, but it's only a partial inhibition. So there's something special about DMT that is not true for DOI or for LSD. So DMT is protecting against this Anna 12. So to a degree, to a partial level. So you're seeing some inhibition. So something is going on there where DMT is somehow related to this TRKB, BDNF pathway, but they could be acting through something else as well. And this is where that evidence is starting to push things.
Did you say that Anna 12, I looked up that it was an antagonist. Did you say it was competitive or non-competitive?
I found non-competitive.
Non-competitive. Then I don't have an answer for you.
Yeah. I found that pretty strange too. So for B, C, again, Scholl analyses, same thing. They're just quantifying what we've already talked about. The addition of Anna 12 in D, of course, you're seeing there's no changes. That makes sense. I mean, this inhibition really works. Again, E, the exact same thing. They're just like hammering it home, And then they added Anna 12 with BDNF and Anna 12 just to show that Anna 12 really does knock down the effects of BDNF. And sure enough, shocker, it does, because that's exactly what it's designed for. So yeah, of course, you're going to see decreases in BDNF with the addition of Anna 12. So do you want to move on to Figure 5 then?
Yeah, sure. I mean, Figure 5 is much the same as Figure 4. They're just looking at the effects of psychedelics on dendritic arbor complexity, as they say, and it's blocked by this new inhibitor, rapamycin.
Yeah.
So one downstream pathway and a very critical pathway is this mTOR pathway.
Absolutely.
So
If you want to build some muscle, you got to get that mTOR.
yeah, I'm sure you probably already have videos on it.
I
You
do.
probably have an entire section on your channel on mTOR.
But it is true that mTOR is in every cell. So go ahead.
So essentially, mTOR is a downstream effect of this TRKB receptor. So by inhibiting mTOR, what they were hoping to show, and what they do actually show again, is that you can diminish the effects. You can, I don't want to say, once again, I don't want to say diminish the effects of these drugs, but if you have the mTOR inhibitor, then you're not going to see the dendritic crossings. You're not going to see the higher level complexities of the dendrites when you do treat with a drug.
Yeah, exactly. Yeah, so rapamycin, as you mentioned, it inhibits mTOR. That's what it's designed for. And what's really interesting here is that DMT, which was the special one in the last figure, suddenly gets knocked down, so it doesn't have the protective effect. LSD seems worthless at this point because it also doesn't have a protective, like it doesn't work anymore in terms of increasing dendritic complexity and things of that nature, which it didn't do that when you knock down TRKB either. So if you knock out either one of those, LSD's out of the picture pretty much. DMT, if you block TRKB, then it's still going to have some effect. But if you block mTOR, it's going to have essentially no effect. But suddenly DOI comes out of nowhere like the champion and decides, well, I'm going to still have some effect even if you block mTOR, which is this master protein synthetic regulator, which is just so, I mean, it's really cool. I mean, you're talking like, this is like signal transduction kind of trying to figure out, what molecules are working on what and things, which I thought was really cool. I started drawing like schematics, trying to figure out, how this could be working, because you're working like a detective trying to figure it out.
Yeah, I mean, I don't know enough about mTOR.
Or obviously these drugs and how they affect mTOR and that whole pathway.
Yeah.
I couldn't come up with an explanation as to why if you knock out the receptor, you knock out DOI activity.
Yeah.
But if you knock out the pathway downstream of the receptor, you regain DOI activity.
Yeah.
Because that receptor, obviously, it's critical for any signaling pathways that come off of it, regardless of how many there are. So if you're blocking the receptor, how are you still getting something else that's happening?
Well, I'll take a stab at explaining that later in the conclusion section.
Okay.
I'm going to skip B, C, and D because they are literally, again, quantifications of exactly what we just talked about. So now let's talk about the final figure. Do you want to take a stab at this? This is serotonin. So do you want to jump on that a little bit?
Right. So 5-HT2A. Right. 5-HT. Anytime you see 5-HT is
Typically referring to serotonin.
Yes.
And there's different receptors and such. So these are rat neurons at this point, plated down again.
Yeah, it's a good point. Thanks for pointing that out.
Yeah, of course. So what they are looking at is they add an antagonist to serotonin. And you can see, in the first panel, figure B, you don't really see anything as expected. And then when this serotonin receptor antagonist is added to DOI, you see a knockdown back to the vehicle levels. And
Right.
you see the same thing with LSD and DMT.
Yeah.
So when you are blocking the serotonin receptor, these drugs aren't effective.
Yeah.
We had an understanding that all these drugs are serotonergic drugs, so they do affect the serotonin system.
Yeah.
And this is just farther evidence of that.
Yeah,
essentially.
absolutely. Yeah, which is really interesting because of something that they mentioned earlier and something you mentioned earlier, that a lot of these antidepressants are focused on serotonin. Yet, 1/3 of the population doesn't get a benefit from those antidepressants, but these drugs work through a similar system, but they are effective. But or at least they're comparable to like ketamine, ketamine is the one that they actually measured that is effective against depression. So really interesting.
So one thing that they did do, and they didn't include this was in the supplemental figures.
Oh, good. Nice.
So in this sentence, this neurite outgrowth, this is back in like one of the first couple
Yeah,
paragraphs. So the structural phenotype seems to only be induced by select compounds. So they actually ended up adding serotonin and amphetamine, like actual serotonin and actual amphetamine to some of these neurons. And they don't see any effects on neurogenesis. So they're not seeing a higher order complexity. They're not seeing, more dendrites being formed. They're not seeing that when they do add serotonin and when they do add amphetamine. But if you change an atom or two and you give them psilocybin, or something along those lines, you are seeing this dramatic effect. So it's really interesting that serotonin, obviously it's native in your brain, endogenous, it doesn't cause the same effects that these drugs do, even though when you knock down the same receptor, you are seeing a decrease in what these drugs can do. So 2 molecules of serotonin, let's say LSD, acting on the same receptor, just like we were talking about in the last figure, yet you see dramatically different phenotypes from the get-go.
Yeah, it's really interesting. I mean, these drugs, there's a lot of similarities, but there's also some things that they're just really strangely different in particular ways. For CDG, same deal. I'm not gonna harp on that because it's again quantifications. They're adding, and you mentioned this earlier, the serotonin receptor inhibitor, the ketanserin. I don't know how to pronounce it, but and then they had various concentrations for H. They tried different concentrations of LSD and then different concentrations of ketanserin. And pretty predictable outcomes there. So nothing completely out of the ordinary there. And that's it, like in terms of the figures. Now, I'd like to wrap this up, but I want to jump into a bit of like the conclusions. What conclusions we can make out of this? Like, what did you think was interesting, and then what do you think that we can make any conclusions out of the data that we've gone over?
Right, so I think that this paper is a good jump start. Once again, without any long-term data, it's way too soon to say, hey, let's all take LSD and be happy,
Right.
However, I do think that the fact that we are seeing this increase in neuronal plasticity, they mentioned, antidepressants, we didn't know how antidepressants work. We really didn't. We've just been using them for decades, and we actually had no idea why they work. But what we are starting to suspect over the past 10 years is that they have something to do with this neuronal plasticity, this ability to kick neurons out of atrophy.
Right.
And what they essentially show in this paper, especially the first couple of figures, is that some of these psychedelic drugs, LSD, Shrooms, they, just like ketamine does, they give these neurons a kick in the butt, and they really start to increase the plasticity. You see dendritic growth. You see synaptogenesis, the ability for new synapses to be formed, more communication in your brain. That's consistent with some MRI images, actually, that they've taken previously of like a guy on LSD, and you just see his brain is lit up, essentially, because you're seeing all these new connections that aren't made, especially in somebody who's depressed. So I think that the big takeaway from this picture is that a lot of drugs similar to ketamine, like psychedelic drugs, can be very helpful moving forward in the clinic if we can show that they actually have efficacy, if we can start to get more papers along these lines of maybe some like human data, something along those lines. So I think that this paper is a good starting point. And there's been, a couple of papers before this that show LSD bound to serotonin, for example. Like they've shown some other things. But I think that this paper particularly gives us a very good start and shows promise of using these drugs in the future.
Yeah, absolutely. I think the main point that I absolutely agree with is that we need long-term studies. We need studies in humans. We need long-term studies, and we need functional outcome studies. That I feel like I say that every week, to be honest, on Physionic. It's just like the molecular things, like it's all really interesting, and it tells you a lot of information. It tells you how to attack an issue. But in terms of does it actually help? Like they reference ketamine, they reference what ketamine has done in the past, what it's going to do in the future, how it helps with antidepressant, and then they have a few data points where they compare ketamine to these drugs, and ketamine still kind of outperforms them slightly, but still, like, it's a really powerful way of showing that what they were referencing in the past applies to the changes that they're seeing in the cells here. But do they actually see, hey, take LSD, see how you feel, and then 6 months later, let's test afterwards? And they're not doing that. They're just showing cellular changes. So can we say there are cellular changes? I mean, no doubt. There are massive changes that happen in the brain. No doubt about that. And again, something we'd have to clarify in humans. But of course, you mentioned the MRI images, and that's already starting to get into that area. And if I'm frank, it's completely speculative, but let's be real. You start taking drugs, you're going to be changing things to your brain. And it seems like, at least if we were to relate the complexity of these neuronal changes to positive outcomes, antidepressant, things of that nature, anti-anxiety inducing, then this is a great way to go, at least based off of this paper. So some really interesting stuff. And I did want a quick jump on this molecular signaling thing that we had touched on earlier. So with mTOR and with TRKB. So because I wanted to jump in on that and give my 2 cents. So I'd written down like a signaling pathway of what could be happening. So what we saw with DMT is you saw a decrease with DOI and LSD if you block the TRKB receptor. If you block that, which is the BDNF related receptor, then LSD and DOI just plummet back down to baseline levels. You basically don't see much of an effect. But with DMT, you did see still a partial effect. So it was partially working through that same system, but it was also probably working through something else. But then interestingly, when you look at mTOR, which is within the cell, which is a master regulator of growth, essentially, you saw that DOI was the one that was still functioning. If you left TRKB alone, you activated it through TRKB, and then it got to mTOR, the signaling molecules attached to one another, and they communicated, and then they got to mTOR, and they were like, grow. And then they had added this drug to stop mTOR from growing, to essentially deaden, to put mTOR into a coma, and DOI was still able to have some expression, some growth of the neuron itself. So what this implies is that DMT has an independent pathway aside from TRKB. So TRKB could be bound by DOI and LSD, especially LSD, and that nullifies its effects. However, if you were to bypass that TRKB, so if you left TRKB alone, the receptor, then you could still see effects on mTOR with DOI. So if you were to block mTOR, there's still something that DOI does that's independent of mTOR, which is really cool, because a lot of people think about mTOR as this master regulator. It totally is, but there must be other mechanisms to tell the cell to grow independent of mTOR, and I have an article on that. So you should check that out, because I do talk about some mechanisms that completely bypass mTOR. So, and this would be a prime example of a drug or just a molecule in general that can bind and go through the same signaling, get to mTOR be stuck just like all the other molecules, but then have another gig, like it's moonlighting, on the weekends or something, and it's finding its way around mTOR to still stimulate neuronal growth. So DOI is not mTOR dependent, or it's partially mTOR dependent, and DMT is not TRKB dependent, so it is only partially TRKB dependent. That would be based off of this evidence. Now, of course, we would need more evidence to really make sure that that's absolutely the case. But I want to throw that in there for any cell signaling geeks that might be listening, which is probably no one. I may have just outed myself a little bit. Beyond that, yeah, so it was a really interesting paper. I learned a ton. I didn't know any of this, literally nothing. So learning this. So thank you for bringing this to my attention, just like you've brought it to like others to my attention, like the BCAA paper and the marijuana paper. Like those were really, really interesting papers too. Like especially the BCAA paper was a killer. That was really fascinating. So add this to the list. So it seems like, based off this evidence, that we can say there are definite changes to the brain. If they were to relate to antidepressant factors like ketamine, which they do try and make that relation, if those changes that they show here do relate long term, this could be a way for people to get out of depressive episodes in a powerful, controlled manner, kind of with microdosing, for example.
Mm-hmm.
That was a great point that you put forward. And not only that, you have this system that is more effectual. So antidepressants, SSRIs, may not be the way of the future. And in reality, we may start looking into something like this, the chemical structures of these molecules, to see how that goes into the future. So is there anything else you wanted to add?
Yeah, just briefly touch on that. One thing, and this was an LSD paper I read a couple of years ago, they think, or theory behind LSD, is that what it does, and I don't know the neuroscience behind it per se, but it blocks a filtration system in your brain, so that everything is now everything that you typically drown out is now being pushed to the front, and your brain doesn't know how to handle it, and that's what kind of produces these hallucinations. So the idea would be, and this is something similar to ketamine, would be to develop analogs of LSD.
An analog would be, what is that?
So develop an LSD-like chemical, like
right.
an LSD-like molecule,
yeah.
That would have the same effects, like the synaptogenesis, that would produce, the higher complexities without giving us the hallucinations. Because then it could be, like, ketamine is only available in the clinic. And unfortunately, it's harder to convince people to get up and go to a clinic to get this medicine as it would be just over the counter or something.
Mm-hmm.
Well, not over the counter per se, but by prescription.
Yeah.
So if we can develop powerful analogs that give these same uplifting antidepressant effects of LSD without the hallucinations, so a person could still be functional throughout their day, would be ideal.
Yeah.
Microdosing is a way to go, and based off just some preliminary data presented in this paper, it seems a promising way to go. But it would be hard to get the FDA on board. Hey, give people a little bit of LSD, because what can they do? They can hoard it until they have enough for a full dose, and then they're doing an illegal drug. So it's kind of a game of cat and mouse between researchers and the FDA, as it always is.
Yeah.
But if we can develop some of these analogs, and I think that we're definitely going to get away from SSRIs in the future, and I think that's ideal for everybody, honestly.
Yeah, I think so too. Okay, well, thanks for being part of the Journal Club, and anybody who is listening or watching the Journal Club. Again, I've had comments from people saying that it's way over their head. I get that. Like, I completely get that. That's not the point of the Journal Club. It's to have this kind of freedom to talk in more complexity. Like, that's the point. And that's why you'll have to wait for the Examine series to come out, where I polish this up and make it more understandable for those individuals that want to know the information without, having two geeks, two nerds nerding out over, the science of every single figure. So thanks for tuning in, and thanks to Bobby for stopping by.
Yeah, thanks for having me, for sure.
And yeah, we'll end it here. Have a good one, guys. See ya.